delay. I'm having technology issues because I wanted to show a video of the Amoeba sisters to review transcription and transcription and to move us into translation, which is what we're talking about today. But I couldn't get it working, my laptop. I might try again at the break. But this is the second to last chapter, so we're almost done. So transcription we did yesterday, translation we're doing today, and then next week we're going to do regulation, which is not a whole, there's not a whole lot of big slides on this, and so we will have a good 2 or 3 hours next week to just do whatever you guys want to talk about. And maybe we can finish all of it today and then have all of next week as kind of a review. We'll see how it goes. I think that sounds like a good plan too. All right, so again, when you do, when we're making protein, we start with DNA, and then we do transcription. And what does transcription make? Message RNA, so mRNA. Where does this happen in the cell? What organelle? The nucleus. So this is happening in the nucleus, and then what's going to happen is this messenger RNA is going to go through the nuclear pore into the cytosol, where it's going to find some ribosomes, ribosomes. And ribosomes are sometimes incorporated in the rough ER, not always, but if it's a protein that needs a little more processing, it might go into the rough ER, but it is going to require ribosomes. So ribosomes are going to be required for the next part, and that's where you're going to take the mRNA and you're going to translate it into a polypeptide. Polypeptide. Peptides are just amino acids. So it's a long chain of amino acids, and you can't do this without tRNA. So tRNA stands for transport or transfer, and the tRNA is what's carrying the amino acids. So it's transporting the amino acids, and of course that ribosome there's one more RNA that you guys have to remember, and that's ribosomal RNA. So ribosomes have incorporated within them these rRNAs. So rRNAs are incorporated into ribosomes, and ribosomes are what's going to do the actual translation. So ribosomes are doing the translation, they're taking messenger RNA, and they're using the code of the mRNA to put the right amino acids in the correct sequence in order to make the polypeptide. So this part here, mRNA going into the making a polypeptide, this is translation. So that's what we're going to talk about today is translation. Taking this messenger RNA and making the polypeptide. And of course the polypeptide, you learned this last term, is going to be folded and eventually into making the active protein. But again, we're just making the polypeptide today from the messenger RNA. All right, that was our overview of what we're about to jump into. All right. So again, transcription happens in the nucleus, and we're just taking that DNA to messenger RNA. Whereas translation is the second part. It is taking the messenger RNA and converting it into the polypeptide, which is that primary structure of the protein. Secondary and tertiary structure, that's the folded structure. So we're just making the primary structure. And we're making that polypeptide.
Your microphone's off.
They usually have a couple of choices up here, and they do not. Hello. I feel like I should start singing, and you do not want me to sing. All right, so can you guys hear me? Yes. All right, I'll use this. translation, again, the mRNA is going to leave the nucleus through that nuclear pore and enter the cytoplasm, same thing. The messenger RNA is going to go to the ribosome, and the ribosome could be on the rough ER, but it could be just in the cytosol itself. The ribosome does not have to be a part of the ER for it to translate protein or translate messenger RNA into protein. And there's going to be 3 steps, just like there was with transcription: initiation, elongation, and termination. Some of you saw that. I was trying to put it in my shirt, so hold up. That's not going to work. All right. So messenger RNA bases, they must be an exact match to the DNA. And that makes sense, because if you don't have an exact match to the DNA, then there's going to be something wrong with that protein. And remember, DNA has triplets, mRNA has codons, and the tRNA has anticodons. And so that's how they're going to match each other. So if I have, for DNA, if I have GTA, okay, that looks like a C, GTA, then what's the messenger RNA going to be? C A U. And so this is the DNA, this is called the triplet. The messenger RNA is called the codon. And then tRNA is going to match the messenger RNA, and the tRNA is carrying the amino acid. So this amino acid has to match. And so the tRNA is going to be nope, almost did it. What rhymes with A? Or not rhymes, but you got it. And what's the U match with? A. So this is the same as GTA, but just written in the tRNA. And this is called the anticodon. So the tRNA has the anticodon that has to match the messenger RNA codon. Okay. Serious and scribbles going on today. And remember what the start codon is. Remember, that's the one that I do want you to remember, the start codon and one of the stop codons. So if we look here, the start codon is always AUG. And so codon, that means it's the messenger RNA codon. So the RNA codon is AUG, and what does that code for? It codes for methionine. So methionine is always the first amino acid. And then there are 3 stop codons that we talked about, and the stop codon that I told you guys to remember is what? UGA. That's where I went to undergrad. There's 3 stop codons that will do the termination, but UGA is one of them, and I always found it easy to remember. So it's the only one I'm making you guys remember. So we're going to briefly talk about some mutations, and some of them I touched on them yesterday, but I wanted to have my slides so I can show them a little more clearly. So mutations, if there's mutations in the DNA, then that's going to affect the RNA, which is going to affect the polypeptide, which is going to affect the protein. So we're going to talk about 4. I want to say there's one more. I'll have to look, but there's at least 4. I think there's one more. I have to look at my slides. Missense mutation. We're going to start with that one. And go from there. We're going to talk about them individually. I think it's easiest. So missense mutation. This is the one, so when you have a missense, and they're kind of intuitive. A missense mutation means it's going to change the amino acid. So which one of these is a missense? Here's a missense. So this is the original. And this one, it's telling you it codes for serine. And don't remember this code, but UCA. UCA codes for serine. So if there's a mutation in that DNA, then you could end up, instead of a U, you could have ended up with a C and another CA. So the C and A are still correct, but CCA codes for proline. So that is a missense mutation. It essentially means that amino acid in place. So do you think this is going to affect the function of the protein? Very likely, or maybe not though. This could be an amino acid that really is negligible in that protein. So if that's the case, if this missense mutation is just one amino acid, maybe it's not an important amino acid. The important amino acids are the ones that are at the active sites or the allosteric sites or are important for part of the folding of the structure. So it depends on where this amino acid is and how important it is. So it could affect the function of the protein or it won't affect the function of the protein at all. So it really just depends. It could be horrible, but it could be negligible. All right, silent mutation is the one we're going to talk about next. Silent mutation does not affect the amino acid. So, for example, UCA here again, here's a silent mutation, and UCU is the mutation. So the codon is different, but it still codes for serine. That's a silent mutation. It still codes for the same amino acid. So this will definitely not affect the protein. You get the same amino acid, serine. So even though it's a mutation, it's not a problem. So that's what silent mutations are. All right, so it's missense and silent mutations. Tandem triplet repeats. So tandem triplet repeats disrupt the mRNA sequence. So they are kind of common, not kind of common. You do have tandem triplet repeats in a lot of your genes, but for the most part, they don't affect the transcription translation of that gene. They can be cut out if they're in the way. But some genes have an excess of these tandem triplet repeats. And when there is an excess of these tandem triplet repeats, then that will disrupt the mRNA sequence and can cause dysfunctional proteins if there's an excess of these tandem triplet repeats in the gene. And this is actually the cause for Huntington's disease. So Huntington's disease is, I don't have it on there, but I think it's in the notes of the slides. It's one of those diseases, it's a neurological condition that you don't start seeing the effects of until a person is around, upwards of 45, 50, 55 years old. But it is fatal. So the person will die early. It's after 50 years old. This does run in people's family. It's a mutation that continues to exist through human evolution because it affects the person's health after they tend to stop having kids or offspring. And so that's why it's still, it's not common, I would say, but it is, you still see it in our population because it is after people are having offspring. So from an evolutionary perspective, it didn't affect the survival of the human organism. So, and people can be tested if this runs in your family. Someone can be tested and they can determine whether or not they will have a shorter lifespan. But Huntington's disease is because of these excess of tandem triplet repeats in the Huntington protein. And there's other diseases too related to it, but that's the most common or famous one. Right. Frame shift mutation is another common mutation. So frame shift, think about reading a book. And if you were to throw in extra words or take out words, then it would mess up the frame shift, the reading of that book. And that's what happens with frame shift mutations. Essentially, this first one here, what's going on is what happens is if there's an additional U, so this U was thrown in there, then you started with, so this is the original. The middle one is the original one. Serine, proline, methionine, and alanine. But if you put an extra U in there, it went from serine, proline, methionine, alanine to serine, tyrosine, and glycine. So it completely threw off the frame shift so that every amino acid downstream is affected. Do you think that this is going to affect the protein? Very likely. Frame shift mutations, because they're going to change all the amino acids downstream of that mutation, it's going to be very problematic for that protein. I won't say it's not black and white, won't necessarily, you know, it depends again on where this is in the protein or the polypeptide. But if you add a nucleotide or you take away a nucleotide, so the second part here is it deleted a nucleotide, and it still shifts the frame. So you have a frame shift mutation because of an addition or a deletion of one or two nucleotides will shift and change all the amino acids downstream from that frame shift. That was the other one. Okay, maybe that is all of them. All right. So these, oh, that's the one, nonsense. I found it. Nonsense mutation. I'm throwing it in. You guys should know what a nonsense mutation is, even if it's not on my slides. A nonsense mutation is where you have the sequence C, C, U, A, T, U, G, and then let's say that the next one is U, G, A. That's a stop codon. And let's say that that wasn't supposed to be the stop. So that's what a nonsense mutation is. It basically blocks, because that mutation, it creates a stop codon, and that it essentially blocks the rest of the protein from being translated. So it's terminated early, which again will affect the protein, especially if it's terminated way early. If it's 1,000 amino acid polypeptide, but that nonsense mutation is halfway through it, then you basically cut that protein in half. So that's what a nonsense mutation is. Since it's not on my slides, I probably will not put this one on the exam. But this is one that you should be aware of as doctors out in the field. Nonsense mutations are just another type of mutation that can occur in your DNA and put a person's health at risk. Oh, thank you. It is on your exam. Actually, I don't know if it's on your exam, but it could be. It's not a hard one to remember. A nonsense mutation just causes a stop codon. Hopefully all my questions will be that easy. All right, so now we're going to start on translation, now that we've got some of those mutations out of the way. So again, translation is where you take messenger RNA, you run it through a ribosome, and you make a polypeptide with it. And the 3 steps are going to be initiation, elongation, and termination. You need a ribosome for translation. So the little blue squigglies are the ribosomes. They come in different sizes. I don't care if you remember the sizes. Just know that the squigglies are the rRNA. The whole thing, so the molecular structure of a ribosome is highly complex. It consists of both RNA and proteins. So the RNA is ribosomal RNA. And both the ribosomal RNA and the protein is necessary to make a fully formed ribosome. And they are split. The ribosome will be split in 2 subunits. There's a large and a small subunit when it comes to the ribosome. I think they do have, oh yeah, they have names, 60S, 40S, and 80S. I don't care if you remember the names. But just know that the ribosomes are these structures made up of ribosomal RNA and protein, and they are required for translation. The ribosome subunits bind to the messenger RNA at the vicinity of the start codon. What is the start codon? AUG. And what does it code for? Methionine. Oh, it actually is here. Always methionine. The codon is a 3 nucleotide sequence representing a single amino acid, like we've been talking about. Start codon is AUG, always codes for methionine. And essentially, the ribosome is going to bind to the start codon by matching it up with its anticodon. And the anticodon is on the tRNA. So, I'm going to blow this up down here. Here is the messenger RNAs in pink. So this is the mRNA. And the mRNA, this is its codon here, AGU. And it has to match the tRNA. This upper part is the tRNA. And there's the anticodon of the tRNA, and it has to match. And when it does, then you can add that amino acid because the tRNA is carrying that amino acid. And here is just another one. The messenger RNA is UAC. It has to match the anticodon of the amino acid of the tRNA, and the tRNA is carrying that amino acid in order to grow the polypeptide chain. And this is just again showing the codon sequence, that AUG down there matching to the tRNA. This was from your textbook. And then this tRNA, this whole blue thing here is the tRNA, and it's carrying the amino acid. And the AUG is the start codon of the messenger RNA coding for methionine. All right, so that was initiation. And I will say also, I didn't mention the 5' cap. So remember there's that 7-methylguanosine 5' cap that you find on the messenger RNA. It protects the RNA, that 5' cap does, and it also helps the ribosome identify the mRNA. So the 5' cap, along with the mRNA that's on the mRNA, is necessary for that ribosome to recognize that messenger RNA transcript. Elongation is the next part, and honestly, this is just the ribosome. So the ribosome is doing the reading here. It's grabbing onto the mRNA, it's running it through its little machinery, and tRNAs are coming and adding their amino acid. That's basically it. That's elongation. It's not complicated at all. I know there's a lot of words on here. It's just saying that one amino acid, one messenger RNA binds, matches to the tRNA here, and here is the messenger RNA, and this orange thing in here is the ribosome. So you need tRNA, mRNA, and rRNA for translation. Here's a question. Which RNAs do you need for transcription? For transcription, you only need messenger RNA. Translation uses all three. So messenger RNA, it's just DNA being converted into mRNA. So it's only mRNA for transcription. But translation, as you see here, not only do we have the mRNA, but we have the tRNA that's carrying the amino acid, and this orange guy here is the ribosome. Ribosomes have the rRNA. So translation requires all three. You need rRNA, tRNA, and mRNA. Whereas transcription, we talked about yesterday, only requires mRNA. That's what you're making in transcription is the mRNA. But basically, you just keep bringing in tRNAs and keep growing the polypeptide chain. So it's just that polypeptide chain is gonna keep growing and growing, and that's what the ribosome is doing. And that's elongation. There's really nothing to it more than that. If I could show my video, you would just see it's just growing the polypeptide chain. And this is showing it from your textbook. And this is something I won't specifically ask you about, but remember amino acids. You have the backbone of the amino acids, which is that, you will see this on the board, so I might as well remind you guys. You have the carbon, alpha carbon in the middle, then you have that carboxyl group on the side, the amine group on the other side, a hydrogen, and then the R group, which is that side chain. And you see here, you have to make peptide bonds here in order to grow the polypeptide chain. And these peptide bonds, so you do have to remember peptide bonds, but that shouldn't be hard to remember. Peptide bonds are made between the different amino acids to grow the polypeptide chain. And it is made between the carboxyl group and the amine group. So this carboxyl group here is going to covalently bond with the amine group of the next amino acid, just like this amine group is bound to the carboxyl group of the previous amino acid. So it's the backbone that's creating the primary structure. It has nothing to do with the R groups. It's all about the backbone of the amino acids, the carboxyl group making bonds with the amine group. And that's going to form the primary structure of the protein. Remember, the primary structure is just the linear strand, of amino acids covalently bound together, and those covalent bonds are called peptide bonds when it comes to polypeptides. And this is from your textbook. I'm not even going to bother trying to go through this. Feel free to read it. All right. Termination. So we're already terminating here. And look what we got, UGA. This is one you got to remember. So this is termination happens during translation using these stop codons. So UGA is one of the stop codons, and there's two others. And that signifies the end of the polypeptide chain. And the polypeptide will be cleaved from the last tRNA. And the protein can then leave the ribosome and be folded into its appropriate structure. So termination is just finding the stop codon. And once that stop codon is found, that terminates protein translation. And then, of course, it has to be folded. This is something we talked about in Biochemistry 1. Here's the unfolded polypeptide chain, so it's just the linear chain. The secondary is when it makes the alpha helices or the beta pleated sheets. Tertiary is the bonding between the R groups. And then fully folded, it can be tertiary or quaternary, but it has to be in one of these two to be active. The primary structure is, the unfolded structure is never active. So the polypeptide itself is useless for the cell. You have to fold that thing up before it's going to become an active protein. But this is what we're focused on right now, is making this polypeptide chain. We talked about the folding in the last biochemistry class. So you can worry about folding. You can let me know if you have questions about it, but I won't have questions about folding. This lecture, this term is just about how we make that polypeptide sequence, that primary structure. And another, if you don't like Amoeba Sisters or you're, want to look for another video, then here's one on Professor Dave, transcription, translation. He might go into a little bit more detail, but he's also pretty user-friendly, gives you the major details that you need to know. So definitely watch videos if you like the animations. Oh, and this is something we're going to talk about a little bit when we get to regulation, which I think we're going to have time for today. And so next week will just be study week, I hope. All right, but this guy's here, there are protein factors that are crucial for the proper functioning of the ribosome and ensure accurate and efficient protein production. I just read from my slides for a change. But these guys here, I don't want you to, there's a bunch of them, there's so many. And again, don't focus on all the details, but you should know what initiation, elongation, and termination factors mean. Initiation factors, guess what? They're going to help initiate translation. That's the gist. They are factors, these are various proteins that help the assembly onto the ribosome. Initiation. Elongation factors are going to be factors that are going to help with the delivery of the tRNA. So these are just, it's not as simple as I made it. Like you just need messenger RNA, tRNA, and ribosomes. No, there's other factors involved. And here's some of the names of some of these factors, but don't memorize these. I think there's only one that I have you guys look at, and that is one of these termination. I don't know. I give you one regulation in the next slide, in the next PowerPoint, just to kind of give you an idea of how one of these works. But elongation factors are going to facilitate, are proteins that facilitate elongation, and termination factors are going to be factors/proteins that help with terminating that translation of the protein. So they just facilitate translation. The initiation of that translation, the elongation of that translation, and termination of translation. And don't memorize these names. I think I'll give you one in the next PowerPoint that we'll talk about a little bit more. But you can see all of them have different functions that just help with the different stage that the translation is in. Oh, and also keep in mind, I'm not going to talk about any of these, but there's also modifications that can occur occur in the amino acid residues, and this can easily occur. Again, the factors can help with some of these modifications because some are modified with hydroxyl groups or biotenylated groups or glycosyl groups, phosphate groups, and that just depends on the protein function. So whatever protein is being translated here, whatever function it requires to have, some of those proteins need other modifications to those amino acids. And don't memorize these, but just know that they could be added to the amino acids in order to facilitate that function of that particular protein. So it depends on the protein as to what type of group it might need to be modified with. And there's lots of different types of modifications that can occur. Wow. All right. The rest is kind of review. And I still haven't put the review slides on these, which is why it's stopping there. I keep trying to push forward. Yeah, so is everyone in? Do you want me to finish today? So next week will just be like a free-for-all review session? Okay. So we are going to give you guys a break right now. So we're going to take 10, and then we'll come back and do the last PowerPoint that you're going to find in week 10. And I'm going to try to charge. Here it is. Oh, I did it quickly. Let me do quickly. Travis trained very well. I feel like I'm trained very well. Someone's trained. All right, 8763. sure to get it. But yeah, I'm trying to get my laptop to connect. So, and it is connecting, but that's not picking. Like my computer's picking it up, but this is not. Because we just got it fixed. So that's the don't, yeah, you can't sign up for Google that you might want. So you never know.
And even then, like a lot of sections have like 6, 7 out of 10, and you're gonna go mesh them together.
Just choose one. 7 a.m. to 7 p.m.
My dad would do that to me, like
So,
posing, but he would try to like rip my toes off.
I'm assuming because there's so many people, all of them right here, these like 9 sections, shred them out.
Try to do. And then bigger
Because like when these guys,
resistance.
Right. But you guys, you guys are
We're gonna prove it.
I like 10% of my.
That's 10% of my house. I didn't transform yet. Nothing, bro. Oh my goodness. Leave me alone, bro. I'm trying to get good grades. You can't get good grades from me, bro. I need to sit home.
Yeah, we got so lucky.
When it comes to subdivision, but yeah, so we're kind of
Yeah, I know. 30 minutes right now.
well, I'm so sure, so that's why I'm
I do have it. I turned mine to pro.
That's interesting. I think I should try.
God, yo. Don't lie.
I don't really know much about it.
I'm in that.
I would definitely say.
I'm gonna teach anatomy. I'm gonna talk about bones, and I have no idea. I looked the geometry about like the top of its made. I don't remember.
So what's one thing I should know?
I know that what the humerus and radius and all that is.
That's the only reason I noticed that. Because like, you got to switch yourself back.
Well, maybe you guys will teach me.
I'll switch everything around so I can have you.
So, do you have a question, guy? There's nothing on there. You guys are just One more lecture, we got this. We got this, and then next week, read questions, reread questions. We'll study together next week. But you gotta go. You gotta focus. Oh my God. I know, I'm already thinking, like, can I give you an online class?
They would pay for tuition.
I've been looking at schedule to make sure you take all my
Oh, it's working.
I'll use this.
Hit.
I can just walk around. I do these, as you guys know. I don't sit still. I do not sit still.
Whoa. Why don't you just use your...
Alright, so this is the last lecture, what I just said that no one could hear. So this is the last lecture, so this is, we're all gonna focus really hard for the next hour, for 30 minutes, however long it takes to get through this. And then next week, study, bring questions, I'll bring questions, we'll have a conversation. Alright, regulation, so we need to regulate, or our cells need to regulate the expression of genes. Because if you have an eye, you don't want it expressing the same genes as your liver. And the liver doesn't want to express the same genes as your blood cells or your muscles. And so all of your organs, is what I'm trying to say, all of your cells have the same DNA. So how do they determine, this is going to make an eyeball and this one's going to make a liver? It depends on the expression of genes. In the lab, you can take a cell and you can change its expression by giving it different cues. So you can, take a stem cell and turn it into a liver cell or turn it into an eyeball. It's the same cell, but you're just expressing different genes depending on how you stimulate it. There's a really good segue into consciousness, but I'm not going to do that. All right, so regulatory genes are going to be controlled, so we need to control that gene expression. And again, this is exactly saying what I just said. At any given time, only a subset of the genes of the DNA are expressed because it just depends on that cell and what it is maturing or differentiating. So when I say mature, I really should be saying the word differentiate. That's what your cells do when they change and grow up and evolve. They differentiate into a functioning cell. So you start out with a stem cell and it will differentiate into a red blood cell or a liver cell, a hepatocyte or melanocyte or whatever it's going to become, whatever function it needs to take on. And so there's lots of ways that we regulate gene expression, and these are just some of the many ways listed here. And we're going to take some of these, not all of them, and just look at some of these a little more carefully, starting with the epigenetic level. This is one I was really interested in, along with telomeres. Epigenetics seems really interesting to me because it determines, based on what you eat, whether you exercise or not, whether your grandparents exercised, whether what they ate, determines how your genes are expressed today. So we do come from our parents who come from their parents. So if you're predisposed to certain diseases or health, then it can be related to epigenetics and what your grandparents or great-great-grandparents did. So, and then we're going to talk about a few related transitional level, post-transcription, translational level, post-translational level. So there's lots of different levels of regulation, starting with the DNA upwards all the way to protein level regulation. So we're just going to talk about these individually. Starting with epigenetics. So in the first one, we're going to talk about, we're not talking about HDOCs yet. So we are going to talk about histones. And we talked about this previously. Remember what histones are. The histones are these green balls over here. But they're actually proteins. So those green balls are proteins. So they're called histone proteins. Histone proteins. And the red is the DNA, and the DNA is going to wrap around those proteins. If it's wrapped really tightly, then you can't express those genes. So if you remember, we have these histone acetylases. So you can put an acetyl group on this guy, or sorry, not on the DNA itself, but an acetyl group on the histone, and that will loosen the chromatin. So the chromatin is the DNA packed around the histone. So when you loosen it, you're going to actually be able to get in there and express the genes on that DNA. But if you take off the acetyl group, then it's going to tighten the DNA again around that around the histone proteins, and so you're going to not have good expressions. So that's what I mean when it comes to chromatin remodeling. Where do I have that? So chromosomes remodeling changes the way DNA is associated with those histones. Remodeling, so you can remodel the chromosomes, aka chromatin. Sometimes you use chromosomes and chromatin interchangeably. Chromosomes technically are more packed than chromatin is. But you can remodel the histone DNA, how it's packed, in order to express those genes better. If it's loose, then you can express those genes better, but if it's really tightly packed and super coiled around those histones, you can't get to those genes, they will not be expressed. And the other one we're going to talk about is methylation. So we talked about acetylation. Methylation is another one. If there's methylation on DNA, so if I put and this one's actually on DNA, so if you put a methyl group on DNA, then it'll silence that gene too. So methylation and acetylation. These things will regulate gene expression from an epigenetic level. Epi means above, by the way, above genetics. Just by methylating or acetylating will affect whether or not you can express those genes. And I do have better slides describing this. So this is all called chromatin remodeling. Or sometimes I call it chromosome remodeling. But the better term is chromatin. So the epigenetic level is chromatin remodeling. It has nothing to do with the sequence of DNA itself. The sequence of DNA, that's another way of regulating it. This is specific to how we pack the DNA, and that's considered epigenetics. Before transcription, before translation, before all that, this is epigenetics, and that is exactly that, right? It's going to regulate the gene even before transcription. And this is showing, so this yellow here is an acetyl group, and you put an acetyl group, for example, on this histone, and you're actually going to make the gene more accessible. So acetyl groups are going to make the gene more accessible, whereas if you remove the acetyl group, it's going to make the gene inaccessible. And by the way, these are the things that affect epigenetics. The way what your foods your mother ate when she carried you in utero, so that affects epigenetics. So just the foods your mom ate when you were a fetus during that developmental stage. Environmental chemicals that might be in your environment can be affecting your epigenetics right now. The air that you're breathing, whether it's polluted or whether it's clean. drugs, pharmaceuticals, aging, diet. So all of these things are affecting your genes and whether they're expressed at that epigenetic level.
Are those pictures on there how you hack the chromatin?
Yes. So this first one here is showing the acetyl group is added. So the acetyl group is added to this one, and that makes the DNA accessible. So adding an acetyl group makes the DNA accessible, and so you can activate the gene. Versus this picture right here, I'm glad you pointed out. This picture right here has no acetyl group, and it shows you down here the DNA is inaccessible when that happens. The gene will be inactive because, as it's showing in that pink there, it's just inaccessible. It's too tightly packed around that histone. So acetyl groups, do they make the gene more likely to be expressed or not? Yes, acetyl groups make a gene more likely to be expressed. The lack of an acetyl group means it's less likely or not likely at all to be expressed. And this is all called, well, it's the epigenetic level, but this is called chromatin remodeling. Because it remodels how your gene, whether you have an acetyl group or not, will remodel the chromatin, how tightly it is packed. The DNA is packed around those systems. So methylation was the other one I talked about just briefly a minute ago. So here, methyl groups. I'll just blow this up so you can kind of see. Methyl group can be added to your genes. If a methyl group is added to the DNA, and it is using, it's specifically to the cytosine, I don't care if you remember that, but when a methyl group is added to the DNA, it turns the gene off. That's called gene silencing. So methyl groups turn genes off. Acetyl groups helps turn genes on. So you gotta remember that. Methyl groups silence the gene. Acetyl groups help with their expression, facilitate expression. So they're kind of the opposite of each other. But methyl groups, when you add a methyl group to the DNA, it actually will silence it. And I'll show you an example in a second here. And this is talking about acetylation that we just talked about. Acetylation will loosen the DNA, allowing transcription, whereas methyl groups, like I said here, turn off the gene. So methyl groups turn off the gene. Acetylation loosen the DNA and that allow for transcription to occur.
You have a question?
Yes. Yes.
Previously you mentioned autosomal recessive, and then so you just mentioned how that how does that
So what I meant by that, what you're talking about there, is this thing. So these aging affects epigenetics. So as we age, there's something about the aging process that will cause chromatin remodeling. And so when I was talking about telomeres, that's something a little different. That's about telomeres shortening, because every time you replicate your DNA, you shorten your telomere, and that will cause you to age faster. If your telomeres shorten faster, you're going to age faster. And if we have a good exercise, lifestyle habits, then our telomeres tend to stay longer, but we have poor lifestyle habits, our telomeres tend to shorten quicker. And that will affect aging. So this is actually talking about something really different. It's just talking about how as we age, we have natural chromatin remodeling. And that can be part of the reason we age as well, but we just don't know enough about it. But we will have differences in acetylation and methylation just because we live, 20, 30, 40, 50 years. Our chromatin remodels during that time, and with that remodeling, and what causes that remodeling are the foods we eat, whether we exercise. So those things will affect us. Everything, our lifestyle habits, and again, including what your mom ate, how we age, whether we age in a healthy way or a negative, unhealthy way, all of that will affect chromatin remodeling, which affects its gene expression, and that can benefit us or put us at risk depending on that. But so some researchers are looking at epigenetic level in relation to aging.
Question?
Yeah.
Don't, at a certain time, is it all aging technically bad for us? Like, at a certain point?
Bad?
or not bad, but like, doesn't that mean that we're getting closer to death at a certain point once we like stop growing and we start aging? Doesn't it like our body's slowing down?
Well, there's everything, so this human machine, I will say, is not meant to live forever. I tell my physiology students that I argue that I think this human machine can live until 300. I think 300 is what this human machine is capable of. I have no proof of why that is or how that could happen. But everything we do affects this human machine, just like what you do to your car affects your car. So aging is natural. Cars eventually are going to run down. This human machine eventually runs down. And so everything we do will either optimize this machine or, be neutral, or maybe it'll bring this machine down. But aging I don't think we need to age as quickly as we do, meaning that our bodies shouldn't run down as fast as they do. So in that sense, yeah, I think we're missing something somewhere. And a part of it, I think, has a lot to do with the potential of chiropractors and working with the nervous system, because a lot of it has to do with our nervous stimulation. And when we start losing that neural stimulation, things start breaking down and not working optimally. And physicians are looking at the nervous system, which blows my mind. And we can talk about this and have a conversation, or would we rather finish this lecture? We have time to talk in my class. I know I want to finish the lecture, even though I think it would be fun to have this conversation, just so next week we can use it as a study time. Or play, and we can have conversations like this. Lecture, right? Okay. One of these days, guys, we're going to have fun. And epigenetics is kind of fun. All right, so epigenetics, this is just showing you a methylation. So this one's all going to be about methylation and the agouti mouse. So the agouti mouse mutation, this was back in 2003. I just finished undergrad pretty much by this time. So this was new in my age. For you guys, it's a little old. But this agouti mouse, see this orange mouse here versus the brown mouse, they have the exact same DNA. Exact same DNA. The only difference is methylation of your DNA. The only difference between these two, so the DNA is the same, the only difference is that the normal gray mouse have a mutation that it's going to be better in the next slide. The mutation causes a color change and diabetes, and that mutation is going to be methylation of its DNA. And so the cloned mouse and the normal gray mouse fed different nutrients. The different nutrients were what stimulated this brown mouse to become this agouti orange mouse. So the specifics are in the next slide. So I'm just going to jump over there to make more sense. All right. So in a healthy mice tag, so a methyl group binds to the agouti gene. So in a healthy mouse, this methyl group binds to the agouti gene. If that methyl group, here's a methyl group, binds to that agouti gene, it will silence it. And so there's going to be, it's going to prevent the agouti gene from being expressed. So this agouti gene is not just one that causes obesity, but it actually causes this mouse to have diabetes-like symptoms as well. And it all comes down to whether or not what that mouse eats. So when this mouse eats lots of foods with methyl groups, then those methyl groups will silence the agouti gene, and you will have a healthy brown mouse. However, if that agouti gene is not methylated, then that mouse, essentially, if that diet of that mouse has no methylation groups in it, then that agouti gene is going to be expressed. And that agouti gene causes obesity and diabetes. So again, same exact DNA. The only difference is epigenetics. The only difference are what was fed to these two mice. And one mouse was getting a lot of methyl groups in its food, which meant it's going to be healthy and brown because the agouti gene was not expressed, because here's this yellow, this orange hue, this is red, is the methylation of this agouti gene. But when there's no methylation, then that agouti gene will be expressed. So methylation silences genes. And the reason why I want to say, like, what your great-grandparents ate affects you, they also did the study where they took this mouse, or both these mice. So this is a female mouse. She's expressing the agouti gene, so she has no methylation. And they gave her no dietary supplementation. They just gave her a normal diet. And when they did, most of her children expressed the agouti gene. She does have some offspring that don't express it. But if you didn't affect their diet, you didn't change their diet at all, then it's going to continue to express the agouti gene. And they followed this through multiple generations and found that this was true. However, if you just supplement their diet with methyl groups, then this female mouse actually had children that had silenced the gene. And so this goes to show you the foods that you eat, the foods that your grandmother ate, your mother ate, these are affecting your genes right now. And some are potentially silenced because of what your great-grandparents or your grandparents or your mother ate. So it's not just the DNA that you inherited, it has to also do with epigenetics and whether or not that gene is silenced or not. And silence in a gene can be good or bad, it just depends on the gene and what its function is. Or it might be neutral. Oftentimes it might be neutral. So what you eat, how you exercise, all of this is going to affect your genes, even if it doesn't change your DNA at the DNA level. So methylation is silencing of the gene. All right. Next, now we're getting into more boring stuff. We're going to try to make this as fun as possible. So this is, again, just some science terms just to help you understand regulation of gene expression. So we have cis-acting elements versus trans-acting factors. Trans-acting factors we're already familiar with, as well as cis-acting elements. An example of the cis-acting elements, so this isn't hard, actually, so don't worry about it. It might not be fun, but it's not hard. So cis-acting elements are regions of DNA that bind to trans-acting factors. So a cis-acting element, that would be like the promoter region. Oh, I actually have it written here. Promoter and enhancer regions are examples of cis-acting elements. So the cis-acting element is the region of the DNA. The trans-acting factor is the transcription factor that binds to these cis-acting elements. So they go hand in hand. One of them is the region of DNA, the cis-acting element is the region of DNA, and the trans-acting factor is just a factor that binds to that region. So transcription factors. We talked a lot about these. So these are the examples. I'm going to highlight in yellow. Transcription factor 2. Remember, it binds to the promoter regions of genes. And what does it do? It recruits RNA polymerase 2. So TF2 is a trans-acting factor. The promoter region is a cis-acting element. A lot of sciencey words for today, learning that fun science language. But this picture is showing you here the element, the cis-acting element is the region of the DNA, whereas the trans-acting factor is the transcription factor that binds to that region. So, not fun, but it's also not that difficult to grasp. So, this one we've seen before. We're just going to get through this one today, guys. All right, this one we've seen before, zinc fingers. It has a mineral in it. What mineral is in the zinc finger? I know you want that on the final exam. What mineral is in the zinc finger? So it's zinc. Zinc is the zinc finger. And if you recall, what's important about zinc fingers is this allows transcription factors to bind DNA. So your transcription factors have zinc fingers. I don't maybe like saying all this. Zinc fingers are within transcription factors. So they're finger-like structures that interact with molecules, and those molecules that they interact with are primarily transcription factors. So transcription factors, most of them have zinc finger motifs. Zinc finger motifs found in transcription factors. And what do transcription factors do? They bind DNA and they regulate DNA synthesis. For example, we've talked a lot about TF2s, transcription factor 2s. Like in the last slide I told you, they help recruit RNA polymerase. If transcription factor 2s don't bind to the promoter region, you can't recruit RNA polymerase 2, which means you will not express that gene. So you need transcription factors in order to express genes. And most transcription factors have this zinc finger motif. And this picture is what it looks like. And that zinc finger motif is really important because without it, it can't bind to the DNA. Sweet. Now, so that was zinc fingers and epigenetics. Now we're going to talk about some prokaryotic gene regulation and compare that a little bit to some eukaryotic. I just picked out some examples, some major ones that tend to be common in gene regulation. All right, so this first one is operons. Operons are only in prokaryotic cells. I don't know if you guys learned about this in micro yet or at all. But operons are found in prokaryotic cells. What are prokaryotic cells? They're bacteria. And what operons are is they are a cluster of genes that are transcribed together under the control of a single promoter. So it's multiple genes under the control of a single promoter. And the reason they're under the control of a single promoter is because they share functions. For example, the most common one that we talk about is the lac promoter. Lac. Lac stands for lactose. So if you have if you have bacteria that are making fuel, so lactose it's a disaccharide, if you remember, and lactose is an energy source for bacteria. And so if they come to lactose, then all they have to do is turn on this operon, this lac, it's called a lac operon, and you transcribe all these genes related to using lactose as an energy source. You don't have to remember what lac-Z, lac-Y, and lac-A do. They're all enzymes, technically, that help you break down lactose and get energy from it. So they all are going to be for the same function, and that's why they're all under the control of the same promoter. And these are called operons. So this is called, literally, it's called the lactose or lac operon. And they're transcribed in a single mRNA, and they're all created together. And that makes sense, especially bacteria. They're really simple, prokaryotic, and so it makes sense for them. Eukaryotic cells, like we have, don't use operons. So this is specific for prokaryotic cells. And this, again, this is also easy, so I hope this doesn't stress you out. But regulatory molecules affect these operons. Activators, guess what they do? They're going to help activate the operon. Repressors, they're going to repress the operon. And yes, I can go into a lot of detail about how and the mechanism of this. I just don't need it. So I'm not gonna. But I do want you to know that these operons can be activated or repressed, which means prevented, depending on other factors that bind to these guys. So other things can bind to these operons. I can give you examples, but I don't want to throw you off. But just keep in mind, proteins and that their activators are going to help control and increase transcription, so activators, versus repressors are going to bind to certain things, and they are going to Okay, it doesn't say it here, but they're going to repress transcription. So activators stimulate transcription, whereas repressors prevent transcription. Oh, that's the only one. There were others that apparently I took them out. Alright, so this is the slide showing you a lot more detail coming from your biochemistry textbook. It makes sense that they put more detail on it. So again, you can repress or induce these operons depending on whether you have an activator or a repressor being bound. But this is the gist of what operons are. They're proteins that are needed for a specific shared function or involved in the same biochemical pathway, and they're encoded together in an operon. And all genes need to use lactose as an energy source, is the example, and are coded next to each other in a single lactose operon. That way they can be transcribed in a single mRNA transcript. And then you can make those 2 or 3 different proteins that you need from that one mRNA transcript. And that, again, makes sense if you're a bacteria or bacterium and you don't have a lot going on. Eukaryotic cells are much more complicated, so operons, just keep in mind, they're specific for prokaryotic cells or bacteria. And this kind of gives you more detail about that if you are really that interested. Okay, so now we're going to be talking about eukaryotic cells. It doesn't say it here, so I'm going to just add it here. So this is, we've passed operons. We're not talking about operons anymore. So this is transcriptional regulation. Transcriptional regulation for eukaryotic cells. And we have already talked about this one. These are enhancer regions, for example, or in transcription factors. So this guy right here would simply be a transcription factor. I'm just going to write TF. And see what's inducing it is cortisol. That's the part that you don't have to remember. But cortisol, if you're stressed out today, cortisol is flowing through your blood, getting into your cells. And cortisol works by going into your nucleus and affecting gene expression. Vitamin D does, vitamin A does as well. These are things like this that affect gene expression and affect a lot of things. And so stress hormones like cortisol can bind to transcription factors and change gene expression. So this is transcription regulation. If you didn't have a stressful day, you don't have a lot of cortisol, you're not going to be expressing these genes. So certain hormones like cortisol, vitamin A, vitamin D, they can go into the nucleus and they can affect transcription factors. And in this case, what you have is cortisol binding to this element. You don't have to remember the element, but it is this glucocorticoid recognition receptor, which is going to sit on an enhancer region. So this is one of the enhancer regions. And remember, when an enhancer region is bound to, it will flip over, bind to the promoter region, and enhance. That's what enhancer regions do, enhance transcription. So, and this is considered transcriptional regulation. So it's related to hormones as well as transcription factors that can activate those transcription factors in order to enhance or promote gene expression. So the key thing here, you don't have to remember cortisol, but I just want you to remember that transcription regulation is carried out via these transcription factors, which are stimulated by various hormones and other molecules. All right, this one we also already talked about, splicing. So this last one we talked about was transcription regulation. This one is post-transcription regulation. So there are 2 different types of regulations. This one's post-transcription, and that's what splicing is. Splicing is, it doesn't happen during transcription, it happens afterwards. That's what the post means, after transcription. And essentially you have the DNA that's expressed into the mRNA, and so here's the mRNA, but yet remember there's exons and introns. The introns have to be spliced out. And oftentimes too, is that you can have alternative splicing. We talked about that last week as well. Or was it yesterday? Alternative splicing, and so here's two different proteins that was made from the same mRNA transcript. And it's just alternative splicing that created two different proteins. That is post-transcriptional gene regulation via RNA splicing. And that constitutes or creates about 70% of genes in humans are expressed as multiple proteins, so a lot of the variability in differences between all of us relate to how our cells, my cells, splice out introns and exons differently than someone else out in this room. And so you can end up with a lot of different variability on proteins just because of that post-transcriptional regulation. So it's the same mRNA, it's just spliced differently. That's another form of regulation. All right, so we talked about epigenetics, we talked about operons in bacteria, we talked about transcriptional regulation with transcription factors, post-transcriptional regulation with spliceosomes. Now we're going to talk about another one, which is RNA stability. And this is also review, because we already talked about these guys, 5' cap and poly A tail. They provide stability. If an mRNA transcript lacks a 5' cap or lacks the poly A tail, it's not as stable, which means it's going to be more likely degraded. It's less likely to be translated. So the 5' cap and poly A tail prevent degrading when the RNA leaves the nucleus. This creates a longer lifespan, and the longer lifespan means that more proteins can be produced from it. That's a form of regulation. And this one's also post-transcriptional regulation. I'm really doing this thing. Are we almost done yet? All right. Okay, here's the one that I didn't finish. It was also from the last slide, so this one's also review. The protein factors that we talked about when it came to translation. So translation, we had all these types: initiation factors, elongation factors, and termination factors. And these are going to help facilitate initiation, facilitate elongation, and facilitate termination. You don't have to know all of these, but I did pick out one. I think it was the EF2 there. So in eukaryotic initiation factor 2, oh, that was the one. EF, so it's this one. This one. EF2. This is the only one I'm having you guys learn. And this is going to be pre-translation. Pre-translation. And what's going on with this guy, if I can try to put it as simply as possible, is that this eukaryotic initiation factor 2, or EIF2, is essentially going to bind to the details you don't need to know, or guanosine triphosphate. I give you the details because it's easier to explain with the details. Guanosine triphosphate here. And this guanosine triphosphate is a high energy molecule, just like ATP. GTP is also high energy. And this is going to allow the tRNA, so it's going to help the transfer RNA. Yeah, let's put it simple. It helps the transfer RNA bind to the ribosome. So that's part of the initiation. In order to initiate translation, you got to have the tRNA and the mRNA come together in the ribosome. And that's what EIF2 is going to do. It's essentially, this is showing it all right here in this picture. A lot of details going in here. Here's the ribosome, here's the EIF2, and here's that GTP, that high energy molecule. And essentially you're going to have these other molecules that are also going to be associated with this come in to grab the, where's the, not that picture. All right, regardless, it's going to help the ribosome scan along the messenger RNA until it finds the start codon. And when it finds that, it's going to initially allow tRNA to bind to that start codon in order to initiate that translation. So, long story short, it's going to help the tRNA bind to the correct mRNA codon in the ribosome. It's going to help the tRNA bind to the start codon in the messenger RNA in that ribosome. And you can't do that without EIF factor 2. It's going to, so it's initiation, so it's going to help the ribosome match the messenger RNA to the tRNA anticodon. And remember, that's going to be how we initiate translation. That first codon is AUG, it codes for methionine, but that AUG and methionine in the mRNA has to be matched to the correct anticodon for the tRNA. And that's what this transcription EIF2 is going to do, eukaryotic initiation factor. And it does this here. So here when you have the EIF2 will only do this when it is, oh, sorry, I was going to say it opposite. And you don't have to remember the phosphorylation, but that's how it's going to do it. This is the actual mechanism of it. EIF2 is going to have to first be, the phosphate group has to be removed in order for that EIF2 to bind to the ribosome. So it's not going to be able to bind to the ribosome in order to match the mRNA to the tRNA unless it is dephosphorylated. I don't care if you remember whether it's phosphorylated or dephosphorylated. All you need to remember is that for pre-translational regulation, EIF2, so that initiation factor 2, has to be connected to the ribosome in order to bring the messenger RNA and match it to the anticodon of the tRNA. I don't know, it's not fun. But it is written out here, and that will, if you want to read through this when you're studying, it'll probably help. But you don't have to remember whether it has to be in this phosphorylated or dephosphorylated form. And here's just showing it from your textbook, a different way of viewing it. So when it's phosphorylated like this, it will not do translation. It has to be dephosphorylated. When it's dephosphorylated, that's when it'll help initiate translation. But without it, you cannot do translation. So that's why it's a regulatory piece. You're not going to translate unless that EIF2 is active. Oh my God, I think I'm done. All right, so this slide here is about chemical modification. We're going to go through some of these because we've already talked about a lot of these. Chemical modifications affect protein activity and longevity, as well as DNA activity, RNA activity. So tell me, methyl groups, do they turn on or silence genes?
Silence.
Silence them, perfect. We didn't talk about phosphate groups, but these will also affect activity, and we have talked about it, especially in Biochem 1. So when you add a phosphate group to an enzyme or take away a phosphate group from an enzyme, that will turn it off or on. It depends on the enzyme. So phosphorylation it just means that when you add phosphate groups, that changes things. It can turn something on or off depending on what it is. Phosphate groups are powerful. Acetyl groups. When you add an acetyl group to a histone, does that relax the histone, allowing the gene to be expressed, or does it condense it and pack it together, turning off gene expression? Allows gene expression by loosening up that chromosome. Exactly. And ubiquitin. When you tag a protein with ubiquitin, do you tag it to be destroyed or do you tag it to be stable? Destroyed. Ubiquitination destroys proteins. When it's tagged by ubiquitin, it's going to end up in a proteasome, and proteasomes degrade enzymes or degrade proteins. So all of these modifications will affect whether or not a protein is even expressed, or if it is expressed, how long is it expressed? And so these are all different ways that your cell can regulate gene protein activity. So what does this other say have? So external stimuli, stress, lack of nutrients, heat, UV light exposure, those will also affect chemical modifications, affect protein activity. Epigenetics will affect things, mRNA stability, splicing, all the things we talked about in the slide. So this is kind of just a little bit of review of some stuff, but you can also ignore it. So this is an efficient way for the cell to rapidly change the levels of specific proteins in response to the environment. So just depending on what's going on, depending on what the cell is, what it needs to do, what the environment cues are telling it to do, it can change things rapidly just by all of these different regulatory processes that we talked about. And there's probably like 100 more we didn't talk about. So I just picked out a few that are really common. So here's ubiquitin. Ubiquitin tags a protein, and when that ubiquitin is tagged to a protein, it's tagging it for degradation. So here it's showing it's been recycled. It's been broken down by the proteasome. So, and that's going to affect the longevity of that protein. If you tag it for ubiquitin, then it won't stick around very long, which means it's going to change that cell. If a protein sticks around for a long time, it's going to stay active doing stuff. If it's degraded, it's gone, then that changes the function of that cell. I might need like a chiropractic adjustment after this. Like this is such a weird like thing going on. All right. So I have 3 PowerPoints that I need to add review slides to, and I will do that this week. So the last 3 PowerPoints, unfortunately, I don't have review slides for them yet, but I'll put those in for the end of the week. Next week, so this week I also have tutoring, CGUS 225 at noon to 1 tomorrow. And next week is just review. We finished the lectures.